Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International GCSE Biology Paper 1BR for June 2022. This is the part 1 video. I will do 2 more videos and put their links in the description box. Let's begin with question 1. It says, the diagram shows a root cell from a plant with structures labeled W, X, Y, and Z. So here they ask which structure is the nucleus. Of course, we can see this one here is going to be the nucleus. Okay, let me get something else. That is the nucleus. We can see this is a vacuole, and then we can see other structures like this one here, part of the cytoplasm. The cytoplasm is here, and this part Y is going to be the cell membrane. So the answer here should be a D. So here they say which structure is not found in the human white blood cells. Human white blood cells, of course, have no vacuoles because that is weird. This is a permanent vacuole found in the plant, so permanent vacuole is not found in human white blood cells. So here the answer should be A. Remember I said X is going to be the cytoplasm, as you can see, and then Y is a cell membrane, and Z is the nucleus. So the answer here should be an A. Moving on. Here they say which molecule is the storage carbohydrate in the root cell. Root cells are plant cells, so they store their carbohydrate as starch. Glycogen is a storage carbohydrate in animals. Glucose is not a storage carbohydrate, and so the answer here should be a D. Next, they ask, the actual width of the cell from P to Q is 125 micrometer. Determine the magnification of the diagram. Here they've given us 1 millimeter is equal to 1,000 micrometer. So remember they've given us this one here, which is the actual width. Actual width is equal to 125 micrometer. So I wanted to convert these to millimeters, which is going to be 0.125 millimeters. So after getting this, and then I use the ruler. You can use a ruler to measure the size of the image or the width of the image. It gave me about 5 centimeters, which is converted into 50 millimeters. Now I have two values that are in the same unit. I can be able to find the magnification. Magnification is equal to image width divided by the actual width, which gives me 50 divided by 0.125, and the answer ends up being 400. So the magnification is 400 times. This brings us to the end of question 1. Let's go to question 2. Question 2. Yeast cells can be genetically modified to produce the proteins found on the outside of viruses. So here they say yeast is a single-celled organism. A yeast cell has a nucleus, and the cell wall is made of chitin. Which group of organisms does yeast cell belong to? So yeast cells are fungi. So the answer here should be a B. They are not animals, not plants, and not products. So they are fungi. That should be a B. The next part says, give a reason why viruses are not considered to be living organisms. Remember, living organisms go through Mrs. Gren. I hope you remember that. They can move, they can respire. Let me get this. They can move, they can respire. Here they respond to stimuli, they can grow, they can reproduce, they can, of course, excrete, and then they go through nutrition, or they can feed. So here I say they do not respire, they do not move, they do not reproduce independently. Remember, they can reproduce in a host cell. They do not excrete, and they do not feed, and so on. Any of these and many more characteristics of living things you would have written here would be okay. So B says a species of yeast is genetically modified to produce a protein found on the outside of a hepatitis B virus. This protein is used to make vaccines to prevent people being infected with hepatitis B. The gene for this protein is inserted into a plasmid, and the plasmid is then used to modify the yeast cell. Give the role of 2 named enzymes used to produce plasmids containing the gene for the hepatitis B protein. Remember, they use restriction endonucleases or restriction enzymes, as well as ligases, which are enzymes that can seal or join different DNA strands or parts of DNA. So here I say the restriction enzymes are used to cut DNA to obtain the desirable gene, and also to cut the plasmid to allow for the insertion of a gene. Remember, they cut the plasmid to create space where the gene that has been cut from the chromosome or from the DNA is going to be inserted. So I said for the ligase, they join the obtained DNA into the plasmid DNA. It's kind of like genetic glue. It glues two segments of DNA together. That is ligase. Here the next part says, the diagram shows an industrial fermenter that can be used to grow large quantities of genetically modified yeast. We can see this is a fermenter. It has a tap down here. There is where air comes in. Air is filtered before it goes in. There is a cooling jacket. Water enters in here and then it gets out here. The air gets out of here. We can see this is a motor for stirring. There is a cooling jacket. There is a paddle for stirring as well. And then a temperature monitor. The temperature monitor is connected to a cooling jacket, which should be telling you that if it senses the temperature increasing, then this top opens so that water goes in order to cool and prevent the temperature from going beyond what it should be. So here they ask, explain the function of the temperature monitor and the cooling jacket. Remember here they've said function, it's singular, not function. So we can collectively take it together. So the temperature monitor and the cooling jacket are there to monitor the temperature, ensuring cooling water comes in to prevent overheating from the heat released during fermentation. Remember, fermentation is going to release heat. That heat is going to increase the temperature of the contents inside. If that temperature is not controlled, the enzymes are going to be denatured and the process of fermentation is going to be prevented or is going to be stopped. So I went on, this ensures that the optimum temperature is maintained so that the enzymes do not denature. Remember, if they do denature, then fermentation is going to stop. The next part, they say, explain why air is needed in the fermenter. Air provides oxygen for aerobic respiration. So this is important. Oxygen is required by these organisms that are inside. Next says, explain why the air is filtered before going into the fermenter. Filtration of air removes any microorganisms, some of which could be pathogenic, from contaminating the culture. So, because if they do contaminate the culture, they are going to compete they're going to compete with the microorganisms that are of our interest. They'll compete for space as well as nutrients. And some of these microorganisms can produce toxins that could affect our microorganisms that are working during the fermentation process. So that could be a disadvantage. This brings us to the end of question 2. Let's move on to question 3. Question 3, the diagram shows part of a food web from an ecosystem in a region of North America. So here we can see the producer here as well as that, the primary consumer, and primary. Here we can see secondary, and so on. So they ask which of these organisms in the food web is a primary consumer. So of course, I already told you primary consumer, we have a mouse, a beaver, and an elk. Among these ones here, we only have a beaver, so the answer should be an A. This one here is a secondary consumer, this one here is a producer, and this one here can be secondary or tertiary, so we can leave them out. The next part says, state what is meant by the term ecosystem. An ecosystem is a group of organisms living together, interacting with each other, as well as with their environment. So in their environment, of course, there is the biotic as well as abiotic components that these organisms are interacting with. That makes an ecosystem. Moving on. Wolves were hunted in this region of North America until they became extinct in 1926. The extinction of wolves also damaged the ecosystem in these ways. So number 1, the population of mice, hawks, and beavers decreased. Let us go back slowly. So when these were removed, the population of mice, the population of hawks, and the population of beavers decreased. Of course, let's see. If you remove these ones here, it means these and these will not have predators. So that gives them a chance to thrive and increase in number. But remember, the coyotes are feeding on the beavers as well as the mice. If the coyotes increase in number, they're going to eat a lot of mice and they'll eat a lot of beavers. Eating them a lot will deprive the hawks from their food. The hawks will have less food to eat, and therefore their population can decrease. Also, we know that over time, so these are going to decrease and these are going to decrease in number. That is going to affect how everything is. And we expect that also the elks are going to increase in number because they have no predators. And that means they're going to be eating more of the grass as well as the aspen trees as well as the grass. And that will mean the producers are going to decrease as well. So that is how that can be. Now, I'm going to take you back to what I wrote. I said, The extinction, the other part, sorry, let me finish here. The population of producers species decreased. I think I've explained why. You remember, once these ones here increase in number, they will eat a lot of this and a lot of that. Once these ones increase in number, they will eat more of this and more of that. That means these are going to decrease, these are going to decrease, and because these have less food, they will also decrease in number. So that is what the question is telling us. So they are going to say beavers are important in ecosystems because they cut down all trees and create shelters that other organisms nest in. Discuss why the extinction of wolves damaged the ecosystem. So I said, the extinction of wolves, which are predators of coyotes as well as elks, led to an increase in the number of coyotes and elks. So the many coyotes ate more mice and beavers, while the many elks ate more grass and aspen trees. Without many beavers, the older trees are not removed, and the younger trees are shedded, meaning they will not get enough sun exposure. So there is less photosynthesis, so the producer population decreased, and there will also be less tree growth that could also have led to soil erosion and fewer habitats for the organisms like birds. Here for the birds, you can include the hawks. So the hawks will have no food for these ones here, less food, as well as less shelter because they do not have enough nesting sites. So that means they can also decrease in number. Let us move on. Here they say, in 1995, 14 wolves were reintroduced in this region. The diagram shows the change in wolf population from 1996 to 2016. So they ask, calculate the percentage change in the wolf population between 1996 and 2004. 1996, we can see it was 50, and 2004, we can see it was 180. So the percentage change should be the change that has occurred divided by the original value times 100. So 180 minus 50 divided by 50 times 100 gives us 260. So the percentage change is 260%. The next part they say suggests why the wolf population changed between 2004 and 2012. The change could be this change here, why the decrease. It could be due to disease. It could be due to competition. It could be due to disaster and so on. So I say there could be disease that killed some of them. There could be competition maybe for food as well as shelter. It could be due to loss of a habitat. It could be due to hunting. Remember previously they were hunted to extinction. So there is a possibility that they're being hunted again, or there is a possibility that some of these migrated to other regions. So one of the, because this is the 2-mark question, 2 of these would earn you the marks you're looking for. Moving on, the reintroduction of wolves caused changes in the population of some plant species. Describe how the population size of a plant species in the region could be determined. We determine most of these are plants, we can use a quadrat. So use a quadrat and remember to place it randomly over the whole area, and then count the number of plants found in each quadrat per throw, and then we have to repeat for several throws and then calculate the mean, and afterwards we can use the mean to scale up to determine or estimate the number of those specific plants within the whole area. So this brings us to the end of question 3 as well as the end of this video. Thank you for being with us. Please do not forget to subscribe. See you in the next video. Bye-bye.